Now I'd like to walk through a few of the exercises in Lab 1 of the manual. On your own, I would like you to complete the information on page 2, where you'll practice converting some metric units from one unit to another, and you may have to measure the length of your finger or something with a ruler in centimeters. I think you can do that just fine on your own. You can skip number 5, which involves the use of a balance to find the mass of an amount of water. And you can skip number 9 and 10 since we're doing this online. Check out part C, which shows you how shows you what different graphs look like and what kind of graph you would use in different situations. And then you can move on to scenario 2. We're going to skip scenario 1. And in scenario 2, it shows a graph, and it says number of new bacterial colonies, and it has days on the, so we have on the x-axis days and on the y-axis number of new bacterial colonies. And it says for number 1 above, write a title sentence for the graph. Start your sentence with figure 1. So that's going to be where your title goes. Just to clarify, we never just turn in graphs in Bio 1. Instead, we turn in figures, and a figure is a graph with a caption. Okay, so if you don't know what that means, you can go to page 77 of your lab manual, and there's an example graph and caption there. So the caption starts with a title. So we never put a title on top of a graph. We always start with a figure caption beneath the graph. We start it with the word figure. And then if it's the first figure of your homework, it's figure 1. If it's the second one, it's figure 2, etc. And then it says for number 2 above, write a sentence that describes the major trends of the data and provide a quantitative description of those trends. So what we're saying there is, what are the major things that happen here? If you had to write about this graph, what are the major things you'd tell me about? First of all, you wouldn't have to describe the change from day 1 to day 2 and to day 3, and then from 3 to 4 and 4 to 5. You describe the major trends, so that if you were to read the sentence and draw what you've written, you get the major shape of this graph, okay? So the major trends are the changes from day 1 to day 3, and the major trends also include the changes from day 3 to day 5, okay? So the data increases and then the data decreases, right? And we can see that on the graph. But you want to quantify that as well. You don't just want to say it goes up and it goes down. By how much? And one of the ways we quantify data in class is percent change, which you can see the formula down here, y2 minus y1 divided by y1 times 100, or fold change, and there's information on this on page 78 of your manual. Okay, for a lot of instructors, if the percent change is greater than 100%, they'll ask you to use fold change in your caption. So check with them to see what exactly they would like to see. But when we see, for example, y2 minus y1, what that's telling you is on the y-axis. So y2 is the one furthest to the right here. So this is the y-axis data for day 5, right, which is 4 new colonies. And y1 is the data here on the y-axis for the first data point, which is 2 colonies, right? That's what we're looking at there. Okay, so let's have a look then on the next slide. So what I would have you do then is, after you complete 1 and 2, is to write a sentence describing the trends of the data, revise that sentence, eliminating any unnecessary words. What I'm doing here is trying to encourage you to write as concisely as possible. Scientific writing is clean, it's to the point, it's concise and precise without unnecessary wording. You know you have a concise sentence when there are no words you can remove without leaving the sentence meaningless. So if you have a lot of extra words in there, now's the time to practice cleaning that up. So once again, I have information in the back of the manual around page 77 on what goes into a figure caption. Okay, there's lots of things that go into a caption. First of all, we start with the word figure, and then we have a sentence that's basically a title. We also have a sentence that describes the changes on the graph, and we include quantitative information there. And there's a few other things we're going to include as well, which I'll get to in the next example. So here is the graph with a caption written. Okay, now what we see here is, first of all, figure 1, the growth of bacterial colonies after 5 days, or over 5 days. Between day 1 and day 3, a 500% increase of bacterial colonies was observed. Now, if you do fold change, that would be a 5-fold increase, but percent change is a 500% increase, okay? And bacterial colonies was observed. From day 3 to day 5, there was a 66.67% decrease in colonies, okay? And then I have some other information here, n equals 1 for each data point. That's something that'll come up more in other figures, but n stands for the number of samples used to figure out each dot. And in this case, we didn't have averages, so there was just one number used for each dot. But if you had to average numbers together for each data point, you would put the sample size there. And we'll do an example of that on another figure. So that's an example of what a caption might look like for scenario 2. Now for scenario 3, I want you to work on it on your own before you watch any more of the video, because if you just watch me talk about it, you will not learn anything. It's very important that you test yourself before you look for answers. Otherwise, you will not learn it well and you will not do well in the course. But scenario 3, you can look on page 5 of the manual, has hours of study, and then 3 different study groups, 3 different groups of students, and their average exam scores, okay? So we have a sample size of 3 in this case because there's 3 different groups of students. And then there's a series of questions. So what you want to do is watch the videos on how to graph on Excel, how to put a best fit line in on Excel, how to write and caption writing and all of that are included on the lab website, and then try to do scenario 3. And after you've tried that, then you can come back to this video and see my answers as an example. Now, one thing that's going to come up in scenario 3 that you may not have done yet, we're going to put error bars in here using Excel. If we were in class, we would graph this one by hand, but I'll go ahead and encourage you to graph with Excel. But of course, your instructor may ask you to graph by hand, so please pay close attention to what your actual lab instructor tells you to do. But when we graph in Bio 1, we're going to often put error bars on there, and error bars can come in a number of types, but we are going to use standard error, or standard error of the mean. And that gives us an idea of how spread out the data are around the averages that we use to calculate that mean. Okay? Now, we can't use those bars to tell if something is or is not significantly different, like one data point to another, but we can say if they're likely to be significantly different. So if error bars do not overlap like these two, then we can say these 2 data points are likely significantly different. If they do overlap, we might say they are likely not significantly different, meaning they're just not that different from each other. Of course, to really say whether or not they are different, we could do a more advanced statistical test like an ANOVA analysis. So here is the figure that I came up with for the data from scenario 3. We have hours of study, we have exam average, okay? And here is the best fit line and the equation of the line. That equation of the line is very important. So hours of study here, that is our independent variable, and exam average is our dependent variable. And you can see my caption here. Now your caption might look a little different. Some instructors encourage you to put some information in the last sentence instead of up here, but all the information that's required is seen here. So figure 1, the influence of study time on exam scores in a Biology 1440 course. Mean plus and minus SE period. Now what is this telling us? The first part here is the title. So the influence of study time on biology scores. There's information there telling me what this graph is showing me. Notice there is no title up above the graph, okay? So this whole thing is a figure. This part is the graph. This part is the caption. And then at the end of the sentence, I have this phrase, mean plus and minus SE. Like I said, some instructors may have you put that at the end in this big thing of parentheses down here, and that's fine. But mean tells me that each dot here is an average, and SE tells me that those error bars are standard error bars, because they could be something else. They could be range bars, for example, or standard deviation, but they're not. They're standard error, okay? And then it says from 1 to 4 hours of study, there was an X% increase in exam scores. So from here to here. Now, you would actually put a number here, okay? You would actually put a value there, and I'll show you how we do that here in just a moment. All right, and then the last part is n equals 3 for each data point. So that tells the reader that for each average here, there were 3 numbers averaged together for each data point, okay? So let's look at some of the answers to the questions on page 5. What grade would you expect from someone who spends 2.5 hours studying? Well, how do we solve that? Well, I have the equation of the line up here. Y equals 8.8333 times x plus 56.5, okay? So y refers to the y-axis, and x refers to the x-axis, okay? So if someone spent 2.5 hours of study, so we know how many hours they studied. Hours of study are on the x-axis, so we can plug in 2.5 for x and solve for y. And so if we do that, we get an estimate of 78.68%. So if someone studied for 2.5 hours on this exam, we would expect they would get about a 79% on that test. Okay, that's how we use the equation of the line. And you can also solve, of course, for y. So number 3 says a student earns a grade of 87%. How many hours would you predict they spent studying? Well, in this case, you set up the equation. We know y. Y is 87%, and instead of solving for y, we solve for x. And if you do that, you get 3.45 hours. So practice this if you didn't get it right the first time. If you look at this and you go, oh, I didn't get those answers, practice it, set it up, play with it, see what you did wrong, teach, learn from your mistakes, teach yourself to do it the right way, and then you'll do well on upcoming quizzes. What is the sample size for this study? N equals 3 in this case. There were 3 numbers used to make each average. And number 5, what is the percent change between the exam average with 1 hour of studying and with 4 hours of studying? Okay, so in this case, we use that equation, y2 minus y1 divided by y1 times 100, if you're doing percent change. And so if we set that up, we can see that y1 is 64.66. How do I know that? Well, let me go back here. y1 is so look at the x-axis, the first value here on the left, and look at what it is on the y-axis. That's y1, and then this one is y2, the one all the way to the right, okay? y2 is the one furthest to the right. So y1's furthest to the left, y2's furthest to the right, in the case of a scatter plot anyway. And so then you plug in the equation there, y2 minus y1 divided by 100, and we get a percent increase of 41.77%. Now, if this came out as a negative number, that would be a percent decrease, but in this case, it's a percent increase of 41.77. So you could put 41.77% increase in exam scores right there, where I have X in the example caption. And once again, I had some, I had numbers here for percent change in the example on page 4, and let me just mention that one more time. So there was a 500% increase, or a 5-fold increase, and then when you, so for this one, this is y1 and this is y2 for the first calculation. For the second calculation, this is y1 and this is y2, and you get a 66.7% decrease. So we explain the increase here and the decrease there. So you may have to explain more than one change in your figure depending on what your data does. You have to use your brain and examine it and decide the best way to describe the data. Okay, so you are going to then end up graphing scenario 4 for an assignment, and you're going to put in a caption and error bars and percent change and all the stuff we've talked about. All right, that's enough of me yapping. Good luck. If you have questions, email your lab instructor.